We ended our discussion yesterday with telophase 1 of meiosis 1. Now, we remember that the stages are the same in meiosis as they were in mitosis. PMAT, prophase, metaphase, anaphase, telophase. The difference is that we had two different stages of meiosis. So during meiosis, it goes under PMAT twice. Okay. If I switch this back to telophase here, telophase 1, you can see that these two are starting to split, starting to divide, each having half of the chromosomes, but still having the sister chromatid together. Now, when we get into meiosis 2, our objectives are going to be the same. Okay? We should be able to identify the stages of meiosis 2. So, on page 12 of your interactive notebook, we have the pictures that we're going to end up drawing into the stages. Okay, so our first objective is to be able to identify the stages of meiosis 2. Our second objective is going to be to be able to determine some major processes that are occurring for those particular stages. Just like on meiosis 1, we had prophase 1, metaphase 1, anaphase 1, telophase 1. We're going to do the same with meiosis 2. So, what do you think the first stage is going to be that we talk about? Prophase 2. So, here we go. In prophase 2, the spindle apparatus forms and the chromosomes condense. Now, one thing to be aware of here on this, okay, one thing to be aware of, look back at your telophase 1. Go back to page, what was that, 10? Go back to page 10 and look at telophase 1. Have we duplicated our genetic material? Did we copy this again? No, we did not. So, there is no duplication of the genetic material. There is no synthesis of the genetic material between telophase 1 and prophase 2. We go directly into prophase 2, and you can see that we have the centrioles in red up there, and they are already navigated themselves to the poles. The nuclear envelope, is it disintegrating again? Okay. Do we have a nuclear envelope there that looks somewhat disintegrated? Yes, but the genetic material has not been duplicated again. Now let me ask you this question. Yesterday we talked about a process called crossing over. Prophase 2, is this where crossing over occurs? What do you mean? I see people shaking their heads. What do you mean? Look right here. Those have crossed over, haven't they? Oh, this is after they've crossed over? Okay. So crossing over did occur, but when did it occur? in prophase 1. So even though these chromosomes have already crossed over, they didn't do it in prophase 2. Crossing over only occurs in prophase number 1. Key fact, remember, key fact. Prophase 1, crossing over. These have crossed over. They've switched. They've been recombinant DNA. However, it did not happen in this stage. Okay, it did not happen in prophase 2. Crossing over happens in prophase 1. Our next phase is metaphase 2, and like all metaphase, what happens? Chromosomes line up in the middle. Metaphase, middle. We call that middle, once again, the equator. So, the chromosomes line up on the equator of the cell on their centromere, much like mitosis. Much like mitosis. So when I show the diagram here, okay, you can see that we have these chromosomes lined up on the equator. We have spindle fibers attaching to the centromere. Okay. We have spindle fibers attaching to the centromere. Now, one difference between metaphase 2 and metaphase 1. Okay? We talked about the homologous chromosomes. Okay? The homologous chromosomes stayed together in metaphase 1. Here, the sister chromatids, okay, right here are two sister chromatids. Here are two more sister chromatids. They are lined up, but now the sisters are going to end up splitting. Okay? They're going to end up splitting. More in a mitosis-like fashion. If I can give you my little drawing, if you remember when we were talking about metaphase 1, we had the X's lined up in the middle as so. And we had the centrioles as so. And the spindle fibers connected and pulled the X's apart, the homologs apart. Is that what's going to be happening here? Are those homologs? No, they're not homologs anymore. The two X's are not side by side like they are right here. So these are the homologous pairs. They're no longer homologs. They're sister chromosomes. So those sister chromosomes are now going to start to spread apart, and it's going to be more like the process we saw when we underwent mitosis. So this is a more mitosis-like fashion, like the slide says up there. So after metaphase 2 comes anaphase 2. The sister chromatids are pulled apart at the centromeres by the spindle fibers, and they are gravitating. They are moving towards the opposite poles in the cell. So the sister chromatid now are splitting. So we're going to see here. Look at our X's. What have now happened to our X's? They've been pulled apart. So our sister chromatid are pulled apart, our X's. So we've broken apart that X, pulling each of the sisters to the opposite pole. Finally, we hit telophase 2. The chromosome reach the poles, and the nuclear membrane and nuclei reform. Cytokinesis, the splitting of the cytoplasm, results in 4 haploid cells, each with n number of chromosomes. So we have 4 haploid cells with n number of chromosomes. For human beings, what would that n number be? What would that n number be? 23. The haploid. Remember, haploid means half. Diploid is 2n, that is double. Remember, D double, H haploid, half. D diploid double, H haploid half. Okay? So the haploid for human beings would be 23. So we have how many haploid cells formed? 1, 2, 3, 4 haploid cells. So, ladies and gentlemen, if we were to talk about this whole process of meiosis, how many divisions did we have? How many times did these cells divide? 2, right? We divided in meiosis 1, and then we divided to meiosis 2, didn't we? And in that whole process of dividing twice, we have reached the haploid cell. All right? Now, remember, we said that what types of cells are produced by meiosis? The sex cells, we got a name for them. What did we call the sex cells? What are they? Gametes. Okay? It's right there. Gametes. The gametes. The sex cells. If it's a male, it's the sperm. If it's a female, it's the ovum or the egg cell. So, these 4 cells that are produced at the end of meiosis are the gametes. They are haploid. They are carrying half of the genetic material.